I'm pleased to introduce my colleague Gordon Taylor, who's a microbiologist, oceanographer, and he's going to talk to us about some of his research in the Cariaco Basin, one of the major anoxic basins in the oceanic basins in the world. And he, along with Mary Scranton and colleagues, have been working in that environment for the last 15 years or so? 16.
It'll be 17.
Oh my gosh, okay. 17 years. All right, so Gordon is got his bachelor's degree at Binghamton University, upstate, and then he went west to Southern California, where he got his degree a little bit after me at the University of Southern California. Then he went out and spent 3 years working with Dave Carl and others at the University of Hawaii before coming here in 1990. So without further ado, you have 50 minutes, Gordon, and counting. Okay. I should also say that lunch today will be in room 113, and along with our visiting students, others that are interested in talking to Professor Taylor or with the students are welcome to join us.
Off to a good start. Sound on? Can you hear me?
Yeah.
All right, great. Let's see how the technology works now. So as Professor Aller said, I'm going to report to you on work that we've been engaged in for close to 17 years now. Professor Scranton and I and a few other folks have been involved here, and I'll expose you to that in a moment. Let's see. There we go. Okay. So a brief outline of what I want to cover today. Motivation for this tropical outpost, why go visit the same place in the Caribbean time and time again. Talk a little bit about the hydrographic biogeochemical setting, the relevance of a time series program in an anoxic basin, why we're there. And then the actual science is going to be in two parts. First, the expression of climate change that we've observed at Station Cariaco over the last 15 years. Summarize, and then some highlights of some of the microbial ecology that our lab has been involved in, some of the exciting findings that we've reported with some of our colleagues. Okay, why the Cariaco Basin? Continental margins and upwelling are important. Marginal seas are a small percentage of the global ocean, about 15%. Marginal seas rather produce about 15% of the global ocean carbon. Upwelling zones are about 1% of the total area of the ocean, but they supply 10% of the total primary production, so their impact is disproportionately large. And more than 40% of the carbon that gets sequestered in seabed is happening along margins. So that was the impetus to set up a time series station on a continental margin, and for other reasons that I'll review, we chose to be over the Cariaco Basin. Here's a cartoon of the bathymetry of the Cariaco looking down towards Venezuela. Its circulation is restricted because of this severe bathymetry. The water exchanges very slowly through these two channels here and here. And the residence time of a parcel of water is on the order of 40 or more years. So the water moves very slowly through this basin. Decomposition of organic matter drives the system to permanent anoxia below about 250 meters. That depth varies from time to time. And the sediments that collect in this basin are essentially undisturbed by critters because there's no oxygen 1,400 meters down, and there's very little in the way of circulation. So the sediments record a very good, high-fidelity sediment record of what's happened over the past. If we look at the basin in cross-section, oxygen goes like this, disappears very quickly, and again around 250 meters, oxygen goes to zero, and then we see the buildup of hydrogen sulfide. This region here, where we go from having some oxygen to none, we call the redox climb because the redox potential of the water is changing from a positive to a negative value. most of the stratification that we see below 200 meters is due to geochemistry rather than physics. This is the density of the water, the blue line here, and you can see it's relatively uniform below 200 meters, so the water is fairly homogeneous in terms of density. And another peculiar thing about the Cariaco Basin, down at 1,400 meters, the water is more than 17 degrees Celsius. So that's warmer than any comparable depth in the world's ocean, so it is warming through time. We can divide the basin into three geochemical regimes. The oxic zone, and the upper layer, of course, is the euphotic zone where we have photosynthesis going on. That light dissipates down about 60 to 80 meters. We don't have much production below that. The redox climb, where we have this transition from aerobic types of metabolism to anaerobic. And then the majority of the water column is sulfidic, hostile to multicellular organisms, but there's plenty of unicellular organisms that love it down there. Okay, so we view the Cariaco Basin as a natural laboratory, and it's useful for a number of questions and interests. It helps us in understanding geochemistry associated with past oceanic anoxic events, and this is just a timeline of anoxia in the ocean. The gray bands are anoxia, and you can see that through Earth's history, the ocean has gone in and out of anoxia many times in geologic history. It also provides an unparalleled sediment record for paleoceanographic conditions over at least the Holocene. It also serves as an end member for understanding contemporary chemical and biological effects of expanding OMZs, oxygen minimum zones. And we'll talk a little bit more about this in a moment. And hypoxic zones. And what happens in those zones is we get altered cycling of carbon, nitrogen, sulfur, to name a few. And obviously, if oxygen is low, it's going to affect the way animals behave and their distributions. Also can provide some insight into evolution of early life in the anoxic primordial soup. And lastly, it provides contemporary observations at local to global scales to compare with proxies that are used in the paleoceanographic record, to interpret the paleoceanographic record. Okay, and this is just an example of a sediment core that's taken through the basin. You can see this banding. Each one of those lightened bands you see represents a season, not a decade or a century, but seasons. So we get seasonal resolution by sectioning through this. And so these core chronologies have been used by scientists both here and elsewhere to compare to ice core chronologies from Greenland and the Antarctic. Tropical treasure trove. You can tell I like alliteration. So I mentioned that the basin is a good analog or end member to study OMZs. The diagram here is published by Palmier and Ruiz Pina, and all of the warm colors represent oxygen minimum zones where there's depletions down to very, very low oxygen concentrations. And you can see that they do cover a fairly large expanse. The Cariaco Basin sits over here, just above Venezuela. But we have the Eastern Tropical, North Pacific, and South Pacific, Bay of Bengal, the Arabian Sea, and there are many other locations. But we don't even have to go that far. In our own backyard, we have seasonal hypoxia in western Long Island Sound, as well as in the Forge River here, just off Great South Bay. So there's pretty compelling evidence to suggest that oxygen depletion is expanding throughout the global ocean. And in fact, in 73% of all published oxygen time series, the trend is negative. Oxygen is going down in many, many coastal environments. What's the consequence? The consequences are many. And this was a nice review paper by Diaz and Rosenberg that shows that when we go from normal oxygen conditions to anoxia, there's a real tipping in the balance of the ecosystem. Energy that goes to mobile predators, so that's the fish and all the things that we care about that we can see with the naked eye. Under normal oxygen conditions, a high percentage of the energy goes to them, and as we have intermittent hypoxia, mild seasonal, severe, persistent hypoxia, the amount of energy going to higher trophic levels, the things we care about, decrease, and all the energy is going to the things that I actually care about, the microbes, 100%. It also destabilizes ecosystems, and that's shown in this diagram down here. From normal oxygen conditions to anoxia, what happens if the oxygen tensions shift a little bit? Not a big deal. The system will restabilize. But as we stress it more and more by oxygen depletion, we get a destabilization. So more energy goes to microbes. There's a habitat compression for multicellular organisms because they can't tolerate anoxic conditions. We get more denitrification, so we're losing useful nitrogen from the ecosystem in these hypoxic and anoxic systems, and we reduce ecosystem stability and diversity. Okay, now to the Cariaco. As many of you in the audience already know, there's several labs that are involved in research at Cariaco. I just want to summarize those for activities for those that are unaware. At Mary Scranton's lab, we're looking at the geochemistry of the basin, its temporal variations and variations in key redox-sensitive elements. And some of the students that have been involved in my lab were working on the microbial ecology, temporal variations, and trying to tease out who the active players are in the anoxic waters, their community structure, etc. And we're starting now to look at functional genes that are involved in nitrogen cycling and sulfur cycling. And then, last but not least, is David Black, who's looking at the paleo oceanographic record from Cariaco. That's David, who couldn't be here today, with some muds from Cariaco. He couldn't be happier. Okay. So now we want to look at the Southern Caribbean Ocean. Our site sits right here. Our base of operation is the island of Margarita. And basically what I'm going to review for you today is, in this section, is a summary of a paper that is under review right now. There's been some recent high-impact papers that have come out based on looking at satellite images and based on looking at ocean clarity, the Secchi depth. Some of you students are aware of what a Secchi depth is. It's the turbidity of the system. And we see fairly troubling titles. Global phytoplankton decline over the past century, climate-driven trends in contemporary oceanography. Both these synoptic type studies say that in general, ocean productivity is going down. But if we look at site-specific time series information, and two examples are here, one from Hawaii, from the Sargasso Sea off Bermuda, and the other in off Hawaii, we see an opposite trend. When we look at time series data, and those have been summarized, 21 different time series have been summarized by Chavez, many of the stations are showing increasing productivity. So we want to know what's going on in the Southern Caribbean, what's happening at Cariaco. And that takes us to our base of operation on the island of Margarita. This is the institute where the marine biology, the marine lab is, which is referred to as EDIMAR. That's the acronym. And EDIMAR actually sits back here somewhere. What is the Cariaco time series all about? We basically started in November of 1995, and we continue to stage monthly cruises. Actually, our Venezuelan colleagues stage monthly cruises, hopefully beyond 2013, but at the moment we're funded to 2013. So at least monthly, the Venezuelan colleagues are going out and making the same measurements that are made at many other time series stations around the world. And these include biogeochemical and physical variables that are sampled. Our group goes down 2 to 3 times a year, usually November and May. We've done this on, I think we've lost count, I think around up to 41 of these expeditions. And the monthly data that's being collected from the ship is complemented by satellite images and moored sediment traps. And that's all shown diagrammatically here, the whole program. Basically, there's a satellite component, the monthly cruises, the moorings that we put out to collect the sedimenting material. Here is our work platform. It's about the same size as the Stony Brook Seawolf, as our own ship. It's not exactly Class 1 oceanographic vessel, but it is seaworthy, and I can't say comfortable. as you may see in a few slides coming up. So what did we learn with our time series, which is shown, an example is shown down here. We know that this, and we knew before the program started, that this is an upwelling system. So here's satellite imagery in the fall, November. You can see the basin right here. The warm colors represent lots of plankton, high levels of chlorophyll. So in November, not much going on, at least in our area. In March, we see lots of warm colors. There's blooms of phytoplankton. What's going on physically? This is a trace of temperature through time, and I've highlighted the 21-degree isotherm. We're using that as our index for upwelling. And you can see that there is a seasonal signal, but there's some irregularity to it. What's driving that seasonal upwelling? It's the annual shifting of the intertropical convergence zone. Our station is right there. It's the blinking sun. And December to May, we get intensification of upwelling, which is driven by the southward migration of the ITCZ. Okay, and for those of you that don't know, the ITCZ is the ascending branch of the Hadley cell. So this is atmospheric circulation. Here's the equator. And so the ITCZ shifts up and down seasonally across the equator. And you can see it's quite asymmetrical due to the uneven heating of land and water and precipitation. This is the kind of records that we're producing. Nitrate, so record of nutrients, record of chlorophyll, and record of primary production. Sediment trap moorings go out for 6 months. They integrate samples every 2 weeks, and then a new sample is taken. I'm not going to dwell on this. I'm going to summarize this in a moment. When we take all of the monthly data that we've collected since late 1995, and we look at the monthly climatology, we look at what's happening with primary production, we see that primary production builds from, you can't read this probably in the back, from December to February, March, then declines throughout the rest of the year. Chlorophyll follows suit. But proxies for phytoplankton mortality, the amount of phaeopigment, which is a degradation product from chlorophyll, actually is pretty level during the bloom and increases after the bloom, as does the export flux, the loss of organic material down towards the sediment. Most of it doesn't reach the sediment, but towards the sediment, is happening in May, so it lags behind by a couple of months. So those were seasonal trends. Now let's look at long-term or interannual trends. And so what I plotted up here is the integrals, total integrated chlorophyll through time, total integrated primary production through time. And you can see that the spikes from upwelling appear to be declining. So what we've done is taken the actual observations and subtracted out the seasonal cycle. So what I've plotted here is the anomalies, the departure from the long-term means, and smoothed it. So the red curve are the smooth data, and run a regression through it to get a rate of decline. And what we see is that over the last 14 years, that the phytoplankton production has decreased at a rate of about 1.7% per year, and the rate of loss of chlorophyll, or I'm sorry, production is chlorophyll 2.8% per year, primary production 1.7, so significant. Now, to your eye, especially in the back, those slopes may have not looked significant to you. So one of our students here in atmospheric sciences, Owen Doherty, who's co-author on the paper, ran bootstrap reanalysis where we considered all the error associated with each monthly measurement. And basically that's telling us that there's less than a 5% chance that these trends are not negative. So they are very robust trends. And essentially from the bootstrap reanalysis, this is the mode. It agrees quite well with what the regressions told us. And basically in less than 1% of the cases where we ran correlations did the correlation between chlorophyll and time result in a positive or a zero zero result. So we did the same thing with primary production, and basically it's telling us that these are very robust trends. Doesn't matter which type of statistic you use, you're still seeing the same thing. Productivity is going down. What isn't changing is the photosynthetic efficiency of the phytoplankton community. Basically, the amount of carbon fixed per unit chlorophyll is rather constant. But another index that has in fact changed is the ratio of the phaeopigment, which remember I told you is a decomposition product of from mortality of phytoplankton. That ratio, normalized to chlorophyll, has gone up systematically. There are jogs and there's ups and downs, but the general trend is that it's increasing. So this suggests that the proportional impact of mortality on the phytoplankton has gone up through time. Okay. To take the phytoplankton analysis a step further, one of our Venezuelan colleagues spends every waking hour sitting at a microscope analyzing which species are there. And these figures basically summarize, each one of these is the annual, each one of the boxes is the annual data bin together. Diatoms used to dominate the system. Dinoflagellates came in second, and coccolithophorids were in close third. Up until something happened in 2004, and you can see there's a downward shift in the abundance of all of these large phytoplankton. So species composition in the plankton has changed. So not just the productivity, not just the chlorophyll, but the types of organisms that dominate the phytoplankton have shifted. Coincidentally, the sardine landings on Margarita Island followed suit, basically the same timeline. These are sardine landings. Again, it's the Margarita fishery, and after 2004, we saw an 87% drop in sardines, and some of the fishery actually has collapsed. People have gotten out of fishing for sardines. So over the last 14 years, we've seen a clear signal that something's happening to the base of the food web, and these changes seem to be propagated up the food chain. These connections need to be explored a little bit more, but the human impact has already been witnessed. This is just a little blurb from the Cariaco website, and basically several fisheries have closed. Prior to that collapse, sardines represented 30 to 40% of the Venezuelan total national catch. So without time series measurements like the ones that our program has provided, smoking gun probably would have been overexploitation. Everybody would point to the fishermen taking too much out of the sea, but we're seeing that their food has also declined, so it's probably both bottom-up and top-down control. What else have we observed? In looking at the data from the sediment trap arrays, we've noticed that rather mysteriously, the downward fluxes of organic carbon, nitrogen, calcium carbonate, opal, which is the frustules of diatoms, it's essentially glass, it's biogenic silica, and the total mass of material sinking down to the trap that sits just above the interface have systematically increased. So production's going down, but export's going up. And that is very puzzling because the current paradigm is that things with heavy tests that have a shell are going to contribute more to the sinking flux than smaller organisms, but that may not be entirely true. So far, collectively, all this evidence is suggesting that there's a state change within the ecosystem around Cariaco. What's causing it? In a reflective moment, we ask. That's the beach right outside the hotel where we always stay, so if you need to get some field work done, come see us. Okay, so let's look at some of the obvious proximal causes. What's happening with the nutrient delivery, nutrient availability? What I've plotted here is the climatology for nutrient inventory in the upper 100 meters. Here we've shown profiles of what's going on with phosphate. So this is phosphate as well. So this is the behavior of phosphate. And if we look at the integrated values of phosphate in the upper 100 meters, you can see, even in the back of the room, I think you can see that this trend is negative and it's highly significant. We're losing almost 2.5% per year of our phosphate inventory to surface waters. Silicate follows suit, shows the same thing. Okay, so why is there less delivery of nutrients into the surface? Well, the thermal structure of this area has changed as well. What I'm showing here is the potential temperature in the upper 200 meters, and there's our 21-degree isotherm, which remember I said is a good index for upwelling. And if we look at the depth, this is the anomalies again, so this is departure from long-term mean. We look at the anomalies of the 21-degree isotherm, we see that it's getting deeper. And you can eyeball that from the graph above as well. So the 21-degree isotherm is not spending as much time in shallower water, it's declining. And sea surface temperature is shown here, and it's actually gone up a full degree Celsius during the 15 years that we've been making measurements. This is the climatology for sea surface temperature, and what you see is that these are the winter months. So we're going from January 1 to December 31 from bottom to top, 96 to 2010, and you can see that there's less winter cooling. So it's not that the summer maximum is getting higher, it's that there's less cooling in winter. So the effect that the thermal structure has is it stabilizes the water column. It causes more stratification, and that's what's shown here. I've basically plotted the timeline for the buoyancy frequency of the waters at our station and plotted the anomaly, and there is a significant trend in terms of stability of the water column. It's just becoming far more stabilized. So what does this all relate to? We can trace most of it. This is basically looking at the annual mean anomalies of these variables, correlating one against the other. Zonal wind, which is the upwelling favorable wind, explains a lot of the variation and everything I've reported to you already. Okay, what have the trade winds been doing, the zonal winds? Here again is the climatology from January 1 to December. And you can see that the warm colors are when the winds are high, and winds above about 6 meters per second are sufficient to get significant upwelling. And you can see these regions are sparser. There are fewer occasions where we're seeing upwelling. And basically this is the zonal wind velocity anomaly, and we've seen that it's gone down at a rate just under 8 centimeters per second per year. So now let's move away from the actual time series station and look at some of the broader scale climatic indices and how they might correspond to what we're seeing locally. And so this is a correlation matrix where we've looked at things like the North Atlantic Oscillation and the Azores high sea level pressure, its location, longitude, latitude, index for intensity of the ITCZ, which introduced a moment ago, the locations of its center of action, longitude, latitude, and then compare that to some locally observed phenomena. And we see that the NAO, in terms of local phenomena, has a weak correlation with our sea surface temperature, but it's the only thing that's explained by NAO. If we look at one half of the equation, the intensity and position of the Azores high, we see that a lot of the variance, the interannual variance that we see can be explained by the position of the Azores high as well as position of the ITCZ. So what have they been doing over this time period? And we should mention that the analyses for the Azores High and our ITCZ are products of Bowen Doherty and Sultan Hamid, and basically they provided me the data. I made plots, and this is what the Azores High has been doing since 1996. it's on a northward migration. Okay, and essentially the Azores High is the descending arm of the Hadley cell, which is shown diagrammatically here, and it's already been documented pretty well over the last 3 decades that the Azores, or the Hadley cell, has been expanding due to global warming, and the IPCC report predicts that the Hadley cell will continue to expand under the present greenhouse gas emission scenarios. So this loop here from the equator northward is in the process of expanding, and there's some migration evident as well. So the ITCZ, just to refresh you, here's another diagram. Our station sits right here. Here's the summer position of the ITCZ on average and the average winter position. From Owen's analysis, we see that the intensity of the ITCZ over South America has systematically been increasing. We see that the center of action for the ITCZ over the tropical Atlantic has been on a eastward and northerly progression over the last 15 years. And if you calculate from these trajectories how much it's moved from 1996 to about 2010, it's on the order of about 600 kilometers. So that whole center of action is moving roughly the way that arrow is pointing. Okay, so these shifts that we're seeing in the ITCZ and the Azores High correspond to the weakened trade winds that we're seeing locally at the Cariaco. Less upwelling, less surface cooling, and less productivity. Okay, and to summarize part 1, what we've seen increase through time is temperatures, degree of stratification. I didn't talk about these. This is a separate paper that one of our Venezuelan colleagues is working on. But the CO2 inventories, the fugacity of CO2 are going up through time, even though this is a productive upwelling system. And our vertical biogenic fluxes have increased. What's going down? Productivity, phytoplankton biomass, trade winds, the density of the surface layer of the ocean, oxygen, which I didn't talk about either, at the lower, in the lower oxic zone is declining. pH is declining. We're seeing evidence for acidification. Surface salinity has declined. Surface nutrients I've shown you, and bacterial production, which I haven't talked about. What's shifted? Plankton community structure, which possibly has led to trophic cascades up to higher trophic levels. So it looks like we have pretty strong evidence that this is all climatically driven and a function of what's going on basin-wide in the Atlantic. It's all being expressed here at our little station off the coast of Venezuela as the Azores High and the ITCZ migrate north in a northerly direction. Okay. So, from the time series, we found that that our observations, direct observations, are consistent with some of the remote sensing papers that have come out recently, i.e. that productivity is declining. Our results are polar opposite to what's happening off Hawaii and off of Bermuda. They're seeing increased productivity as well as increased export flux. And what it points out to us is that there are regional differences that we need to look at more closely. They require more scrutiny. And to make a plug for time series, there's really no substitute for direct observations. So when it comes to understanding local expression of climate change, you really have to have long-term local direct measurements. And basically this reiterates what I've already said, that these locally observed state changes in the Southern Caribbean appear to be indirectly driven by the global heat budget and evident by the migration of Azores High and the ITCZ. Okay, my title said, and other tales. So real quickly, our lab is more involved in microbial ecology, molecular ecology than ITCZ and things, so that's kind of a departure for me. And so what I want to just show you is some really, what I find exciting results from looking below that surface layer. What I reported on is basically surface layer, meaning what's happening in the upper 200 meters, and focus on what's happening in that anoxic basin. And we were fortunate enough to get 2 NSF microbial observatories funded a few years back, and they started in 2004. And our purpose was to go out and characterize the composition and activity of anaerobic communities of bacteria, archaea, and protists, protozoans. For those of you that remember the old terminology. And to get some assessment of how these communities change in space and time, and with some luck, isolate some novel taxa that have very interesting biology. This is a very busy plot, I'll grant you that, and that's why I put those blinking red arrows. I want to draw your attention to what's happening where oxygen goes to 0 and sulfide begins to grow in concentration, as does ammonium. What I'm pointing to in this first panel on the left are rates of chemoautotrophic production. So these are bacteria that function like plants in the dark, except that their energy is coming from reduced chemical energy, from sulfide, other species of sulfur, or ammonium. And basically, they're harnessing the energy in those inorganic molecules to drive their metabolism. We always see an enhanced production of these types of organisms across this transition zone. I've shaded the redox line of the transition zone. We always see enrichments in the numbers of microorganisms that are present. That's this blue profile here for bacteria and archaea. The gray profile is for the protists. These are consistent features. The depths change, the shapes change, the magnitude changes, but there's always a signal. And these organisms seem to do best where the opposing gradients of oxidized materials meets the gradient of reduced materials, which they're harnessing for energy. In our lab and with other colleagues, we've strived to understand the population dynamics, what kinds of organisms are responsible for this productivity, because it's not insignificant. There's a fair amount of carbon that's being produced in this sort of secondary ecosystem. And so we've used molecular probing techniques such as FISH and cardfish. FISH has nothing to do with those things with fins and scales. It stands for fluorescent in situ hybridization. So, fluorescent in situ hybridization. So you basically take a genetic sequence, very small number of nucleotides, and it has a fluorescent beacon, and we hybridize it against the complementary genetic code, and the cells then light up and report back. And that's shown in this right panel. These are probe-positive organisms that had the complementary sequence to our probe, so they fluoresce this reddish-orange. The total community, this is the same microscopic field, we're looking at the same field, using a different stain that stains everybody's DNA. So that's the total community. That shows all the bacteria, and this shows the bacteria that have the molecular probe that we're interested in. So that's how we develop these profiles. And it tells us who becomes important as the geochemical regime changes from surface waters to depth. And I'm not going to spend any more time on it. Just it's an activity that our students spend a lot of time on the microscope developing. The other thing is how complex. I mentioned the comfort of the ship. A lot of our students end up in this position. So what about animals? Well, animals below the interface are probably not too important. There is evidence, very good evidence, that small fish and zooplankton do migrate down into these anoxic waters on a daily basis, probably to avoid predation. But we know nothing or little to nothing about their activity at depth. When they're down there surrounded by sulfitic waters, what are they doing? Just holding their breath, waiting for nighttime so they can come back up and forage again in the surface waters. So the top predators are probably protozoa, and we've measured large enrichments across the boundary from oxic to anoxic, where protozoans are very abundant and, as you'll find out in a moment, extremely diverse. In most ecosystems, we know that these protozoans are living by consuming small algae, bacteria, and other protozoa. So a very coarse conceptual model of the food web in the waters below, or the anoxic waters where the oxygen runs out, is more or less like this. The bacteria are the primary producers in this system. Flagellates are their primary consumers, and there's a nice example right there. And the consumers of the flagellates are probably ciliates, and many of these ciliates can, in fact, consume bacteria as well. We also know viruses are important. We've done studies on the viral distributions. We know they're enriched where the bacteria are enriched. We don't think the zooplankton are playing much of a role, and of course there's no algae because there's no sunlight. It's pitch black, no photosynthesis going on. Okay? So, yeah, here we go. You can all see that in the back of the room, right? Yeah. These are actually two phylogenetic trees. One for the class, for the phylum of ciliates, type of protozoan like Paramecium, remember from high school biology. Euglenozoans, Euglena, you probably remember that from high school biology as well. These trees represent a heroic sequencing effort on the part of colleagues of mine at Woods Hole, where they developed 18S ribosomal RNA libraries that should be familiar to some of you. They sequenced over 20,000 clones. Of those 20,000 clones, 6,500 were clearly protozoa. And they followed up with 454 pyrosequencing of 80,000, which yielded 80,000 different sequences. This is the most, to date, the most massive sequencing of a single environment that we know of. And the interesting thing that came out of these samples from anoxic water were these two deeply branching clades, or groups, this one here and this one here. They've only been observed in the Cariaco. There's no known named relatives in anybody's collection or the literature anywhere. The other interesting thing that came out of these analyses were that this is basically the pie charts representation of different groups of protozoa. In oxic waters, 40 meters above the interface, at the interface, 40 meters below the interface, and then down at 900 meters. These Venn diagrams represent the amount of overlap in the types of sequences that we're seeing in the waters above the interface and the waters below the interface, only separated by 80 meters of ocean that is homogeneous physically, but very different geochemically. The bottom Venn diagram is the interface versus 40 meters above, with an 18% overlap in sequences. So what this tells us is a tremendous amount of endemic distribution of organisms, highly specialized for a particular layer. The other interesting thing that came out of the analysis is when we were lucky enough to have good, well-preserved samples to look at under the microscope, that a lot of the protozoans had symbiotic bacteria all over them. These are healthy protozoa. Each one of these little loaves of bread is actually a bacterium that's doing something beneficial, we presume, to the ciliates, but we have no idea. Okay, so there's a high degree of apparent endemism. That's a hard word to pronounce, but it means, okay. So, a little video for you. And this basically shows one of these novel ciliates. That's his mouth right there. So it's not easy to do video in a scanning electron microscope. But our student at Northeastern was able to do it, Bill Orsi. Bill's now doing a postdoc at Hui. Okay. All right. So this little video that you just saw is of an organism that's only 40 microns long and probably the top predator in this anoxic ecosystem. One of many, not the only one, but one of many. This organism has no known named relatives anywhere in the world and apparently belongs to a newly recognized phylogenetic lineage, which we call Cariaco. This has been accepted by the International Journal for Systematics. We have just named this new class in honor of the Cariaco, and we know nothing about its existence in the anoxic world. I should also say that this is the first new class of protozoans identified in over a century. So we're kind of proud of that. And that's Ginny and Bill enjoying the comfort of the Hermano Gines research vessel. They're all sequenced out. After 20,000 clones and 80,000 pyrotags, they're just exhausted. This organism made it to the cover of the International Society for Microbial Ecology journal. They were the cover girls on this journal for all of 2011. And these are some micrographs of the organism that are in the paper that has been accepted and is in press right now, naming this new class. And these are line drawings that were produced by a classical protozoologist, the guy that sits at the microscope all day long and counts hairs on these 40 micron cells. Okay, well, we're now in the process of taking the data that we're getting from Cariaco a step further, and we're doing comparative studies. I wasn't involved in this study. This is actually, again, Bill Orsi, who was involved in the Cariaco project, has gotten samples and sequences from 2 fjords, so the Saanich Inlet, which is British Columbia off Vancouver, and the Franvarn Fjord. And basically this hierarchical PCA clustering tells us that the community composition of Cariaco segregates very nicely from these other 2 fjords. So it's fair to say that each of these anoxic environments does have its own endemic communities where there are very few shared species. That's what this analysis is telling us. However, at Saanich, they did detect a couple incidents of sequences that fell into the new class of Cariotrichia. So Cariotrichia probably is more broadly distributed, not just a parochial Cariaco resident, but has been around for a long time and is in many similar types of environments. So, from the other tales, to sum up, what we found in our observatory, and this is a really brief summation, we found many other things, but we see that the anaerobic communities are dynamic for bacteria, archaea, and the protozoans. Symbiosis, really interesting, really important, and in some instances we think all 3 domains of life are involved. Protozoan as host, bacterial ectosymbiont living on the outside, with archaean endosymbionts living in the cytoplasm, if they're anything like their shallow water relatives. But we know nothing about what those interactions involve, what the mutualistic types of relationships might be. We've seen an assortment of both endemic and cosmopolitan phylotypes from the massive sequencing efforts, and many potentially new species. And I say, is that a big deal? Anybody that does microbial molecular ecology can go out any day of the week and discover new species. It's like falling off a log. There's nothing to it anymore, because our understanding of the diversity of unicellular organisms is so poorly developed, because we didn't have the techniques until 10, 15 years ago. So discovering new species of microbes is not the same thing as finding new species of a fish or a mammal or something like that. That's hot stuff. New species of microbe, don't bother me. You know, it's not that big a deal. But finding things that branch so deeply in the phylogenetic tree, and we have, I discussed the example for the ciliate that has this clade that's now called Cariaco trichia, but the euglenozoa also have a deeply branching clade that's unique to Cariaco, at least so far. And that has some evolutionary implications, that there is far more deep-lying diversity that's cryptic that we really don't know much about. Oh, this is the wet lab on board the ship. You can fit 3 people in it, but you all have to agree when to turn to the left or the right. So colleagues ask me, why didn't you do this? Why didn't you do that? And I show them a picture of the ship, and they say, oh yeah, I understand. And here's another example of an association of a ciliate. This is an electron micrograph of a ciliate, and it has a different type of symbiotic bacteria living on it, banana-shaped symbiotic bacteria. So it's a very common feature that we're seeing with the microscopy. And with that, I'll close, and I think I made it at 55 minutes, right, Joe? 50.
Thank you for those 2 talks, Gordon. There's time for maybe 2 quick questions, and then Mary, is that fair? Okay.
I was just going to ask if there's any way to tell genetically what those symbiotes might be, because people have reported, say, sulfate reducers or methane reducers as symbiotes in the Black Sea, and that would be consistent with a lot of the geochemistry.
Right. Well, we've talked about using fish. The main problem is preservation. To get those cells in the images that were presented, that, as you recall, they had to send an instrument overboard that actually collected and preserved in situ at the depth, bring it back up. So if you get samples like that, then we can do fish that probe for something like sulfate-reducing bacteria, because there are fish probes that we use all the time for that group of organisms. So you can make them light up, basically, and see them. Preservation is an issue.
Can't you just bulk sequence?
Oh, but then you don't know they're associated. You don't know that they're associated with the host because it's just mashed up DNA. Ellen?
Gordon? So I was predictably interested in the sardine fishery and its collapse. And you talked about how, your studies show that there's a lack of food involved, but you also said that the smoking gun would normally be the fishery. So has anyone tried to look at all those data together and to tease out how much could be fishery induced and how much could be a bottom-up effect?
Not to my knowledge, but fishing effort is tricky, right?
It depends on what kind of data.
Right. And how cooperative your fishermen are.
Yeah, I think it could be really an interesting case to look at.
Bob?
Since the biota seemed to really undergo a change during the 15-year period you've been looking at and seemed to be very suggestively connected to the NAO behavior, has you or anybody else looked back in time? The NAO is well documented by going into sediments deposited at the bottom.
Right.
You ought to be able to
well,
confirm
Sullivan's
this.
been involved with David Black, right? You've looked at, so you could answer the question because I don't know the answer.
Okay.
But I know, yeah.
No, I don't know the answer either.
You don't?
Actually, it's not the NAO, it's the main thing that we found was that the NAO does not have any correlation. It's the high, you see the subsiding arm of the catalyst cell, which is moving, shifting northward. So it's a secular trend. NAO is a fluctuation back and forth.
Okay, but
So,
one might think that over the last 10,000 years, let's say, it hasn't, this is not the first time that it's migrated.
Yeah, so I think so what Gordon's work has shown is that as the headless cell shifts on a very large scale, it has this local strong signature on the ecology. And I'm sure it has happened many, many times. And in the Cariaco literature, people often interpret their the changes they observe in terms of presumed ITCs issued, including on failure timescales.
Right, right. Yeah, so David Black has looked at it.
Yeah.
And I thought you collaborated on that as well.
No, but we didn't find, I mean, on a much longer timescale.
Right, right, right.
One more question.
I told you no questions coming.
This is an easy one. So you say that the amount of production in the surface waters is decreasing, but the export of flux to the deep water is increasing.
Yes.
Where the hell is that material coming from?
Just smaller cells that aggregate. This is not unprecedented. It's been shown actually as a review paper where they've shown that smaller cells that don't have the ballast, that don't have tests in the Arabian Sea, the Sargasso, and the Pacific, contribute significantly to the vertical flux. So it's just that there's probably a shift in the organisms that are packaging the material, and their diet has changed because the phytoplankton composition is changed dramatically.
Maybe less grazing in the source.
So what are the zooplankton populations look like?
Those data are not fully analyzed yet, but
Yeah, there's some problems with the zooplankton data because of the ship they can't do opening and closing nets.
Okay.
So
Yeah.
there's issues of where, we've had some arguments.
Yeah, it's problematic.
Trying to get it published, but there's arguments about whether they're sampling known layers or not. So it's, again, it's a technological.
okay. We're exhausted.
Yeah.